Do you see the correct screen on Zoom? Okay. Okay. Not sure. Probably it doesn't matter for me, but for others. Should I start, Constantine, or is it? Are we ready? Okay. Okay, all right then. Then hello everyone. Again, we're gonna basically continue from where we left from last week. So a couple of slides to remind you what we were talking about. So we started with the quick introduction to genome analysis, and then we were talking about these machines that generate the sequencing data. They can generate the data at a very fast rate, but the machines that we're using, they are general purpose machines, so they don't really know the requirements and the data types, etc. of the data that we're trying to analyze. So that's why they were slow. And then we said that we could also look at different architectures, even systems, the heterogeneous systems, including different architectures to accelerate the genome analysis, even for basically for mobile solutions. And then we said that for that, we need to essentially look at the entire stack of computer architecture to co-design everything. And we essentially showed this pipeline where we have many steps in genome analysis, starting from the data generation up until the further steps in genomics to reveal the answer that we're looking for from the genomic data. And we briefly essentially touched on how we generate the sequencing data, what sort of technologies we have, and we quickly discussed how nanopore sequencing works. And as you remember, we have this biological molecule where we input this to a machine that we call sequencing machine, high-throughput sequencing machine that generates the fragments of the DNA that we call reads. And then there are some challenges to analyze this data. One challenge is now, once we have this digital data, which is basically the representative of your biological molecule of the DNA, we don't know the origins of the locations, right? We need to find them. And sequences, these reads can contain errors. And also we have now many reads to analyze quickly. And we essentially said that we can actually start with the raw sequencing data analysis, where we either base call it, translate the raw data to ATGCs and then analyze it, or we can directly analyze this raw data without translating, which can provide us better, let's say, benefits in terms of performance and energy efficiency. So this is basically where we were last time. And today we're going to basically cover the remaining steps here, these last two steps in genome analysis quickly, and then we will move on to some of the works that we do in our group to accelerate some of these steps here in the genome analysis. So then let's continue with read mapping. The goal of read mapping is essentially, in a sense, to solve life's puzzle from the sequencing output. So as I said, we have the fragments of your genome, we don't know their origin, and they can contain errors, right? So this is basically, I like to basically make this analogy where, so these DNA fragments are like actually the pieces of the puzzle, right? So your goal is essentially to solve that puzzle. And to solve this puzzle, what you can essentially do is you can either compare these fragments, these DNA reads, let's say, to each other, so that you can sort of understand what this picture looks like, right? So when you compare these pictures to each other, the fragments, you may have some understanding of basically what it looks like. Or you may have, like in the puzzles, you may have a template picture that you can look at, so that you can maybe even more easily find where these fragments belong to in the corresponding genome. So we call this template sequence that we use for guidance a reference genome. And actually figuring out where these genomic fragments belong to and then how they compare to each other is very essential so that we can understand the functionality of a sequence, the mutations and the diseases and so on. So basically this sequence comparison is really essential, is at the core of genome analysis. So let me then provide you maybe a bit more realistic scenario or realistic view of it. So this is how DNA, let's say, logically looks like. This is how physically looks like. And at the end of it, when we get the reads from the sequencing output, this is what we have, right? And then what we really want to do is that we want to figure out where these fragments belong to in the reference genome. So this is basically then you have many of reads, and then the size of the reference genome can be huge. And your goal is actually to figure out is to analyze all of them so that you can basically get a full picture of your own genome by comparing it to the reference genome. So what essentially, as I said, the challenge is you're going to have perhaps maybe millions or billions of these reads to compare to the reference genome. And if you are familiar with the approximate string matching algorithms, a naive approach would be you have a fragment, a read, and then you have a larger reference genome, and both of them are strings, right? But reference genome can be of length, let's say 3 billion characters in the case of a human genome. And the read is usually around from, let's say, a few hundred to few kilobases. Now, what you could do naively is essentially start from the very beginning of the reference genome, and then for every character of the read and every character of the reference genome covering the region that the read covers, you can essentially basically traverse the entire area of the reference genome, do your approximate string matching in the entire place, but then this would be extremely expensive. So this is basically the complexity is polynomial. And given that the length of the genome is high and also the number of reads that we need to analyze is also very large. So this is a very basically computationally costly approach, right? Of course, there are practical solutions to it. This is basically what I'm going to cover now. We may make use of several data structures and several heuristics so that we can quickly figure out where a read belongs to in the reference genome. So as I said, this is a reference, and this can be of length 3 billion characters in the case of human. And what we initially do is that we do this step that we call indexing, where you store sort of the contents of the reference genome in a data structure. And in this case, we have a hash table. And this data structure is useful for us so that we can quickly basically query some subsequences so that we can identify whether these subsequences exist in the reference genome, and if so, where they appear in the reference genome. So what we can do is that we can essentially take all or certain subsequences of fixed length, let's say K, or we call k-mer, from the reference genome, and then store their positions, the k-mers and the positions in the reference genome, and then we build the index doing so. Then later on, once you basically want to identify or find whether this read also appears in the reference genome, what you can do is that you also extract the subsequences or k-mers from the read. And then what you can do is that you can simply query these k-mers using the hash table and then figure out if they appear in the hash table, and if so, where they appear, right? So this is basically the seeding step. You sort of seed the query the reference genome using the hash table to find the potential matching regions between the read and the reference genome. And then it may so happen that you may hit also some false regions, because what we're mainly doing is that we're using usually short subsequences here, and then assume that you are using 20 character, 30 character long short subsequences, and then they can appear actually in many regions. So our goal is really to identify what is the most likely region that essentially where this read can truly be coming from within that reference genome. So to identify those, in the next step, what we do is that is a seed filtering step. this is usually done either by an algorithm that we call chaining, where it tries to basically find the collinear matches of multiple seeds if they are in a close-by region, or you can essentially then even perform actual filtering that figures out whether the read itself is going to be highly dissimilar to the region where you have seed matches. Then you perform alignment. The alignment would figure out the exact differences and the matches between a read and the reference genome within the candidate region that we suspect that this read can belong to in the reference genome. And then basically, since this alignment is basically a very sensitive algorithm, which is an approximate string matching, and if you figure out that this read is coming from that reference genome, we say that this read is mapping to that particular region in the reference genome. So essentially now you figure out where this read belongs to, what its origin is, and also what are the mutations, potential mutations we have in that read compared to that reference genome. So we're going to utilize this information later on. And since it is also very important to figure out the seed matches, the matches of these subsequences accurately and a quick way, we also look at this problem where we essentially try to quickly find the seed matches in an accurate way. And if you're interested in this basic problem, you can take a look at this paper. But what we mainly figure out is that once you perform all of these steps, which is basically indexing, seeding, alignment, and then filtering, and so on, what we find is that around 90% of the time we spend for the read mapping, we spend either for alignment or for chaining. So then this basically gives you an idea if you want to accelerate these steps, or if you want to accelerate the read mapping, you initially want to focus on the alignment and the chaining parts. So the second question is, so initially I showed you we've been using a single reference genome, but then the question is, do we really basically want to use a single reference genome? And basically this can be problematic because a single reference genome can be only representative of only a few individuals, right? And then we really want to represent as many individuals as possible so that our analysis is accurate. So this is basically an illustration of the problem, again, using the puzzle solving analogy. So here we have a human reference genome, which is, as I said, a 3 billion character in the case of human, and then we have reads, right? So what we do is that, so your reads are not essentially going to be exactly matching to a reference genome. There are going to be variations. And it may so happen that you can recognize these variations, you can tolerate these variations during your seeding step and chaining and the alignment step, and then you can still map your reads to a reference genome by tolerating those. But if you have a huge variation in your read, then you may miss that read mapping to a reference genome, right? You may basically fail the mapping of a read. And then this can be either due to whether that read is very erroneous, then it may make sense that you don't map it to a reference genome. But then it may also contain a huge variation compared to reference genome, right? So you don't really want to miss that because it is a useful information. And then, but then if there is such a case, then you may miss it because you're using a single reference genome. So then the solution to it would be, okay, let's not use a single reference genome, but then use multiple reference genomes, right? So multiple copies of them. But then this is not necessarily a very clever solution because this is basically going to increase your runtime. If you are using, let's say, 4 reference genomes instead of 1, then approximately you're increasing your runtime by 4x as well, right? So you really don't want to do that unless you really have a good reason for it. The second solution would be actually the use of graphs. And the intuition behind is that if you are mapping your read to a human, and approximately the humans will be highly similar to each other, the DNA content of the humans will be highly similar to each other. So this means that there is a huge amount of data that is redundant or that is similar across individuals. And you don't really want to copy that information or redo the computation for these similar regions. So a graph structure can easily recognize this redundancy, the similarity, and it can also can give you basically an efficient way of also annotating the differences across individuals if basically we have a difference at a certain position, right? So in doing this, we can encode both the similarities across individuals, but also the differences between them. And in doing so, you can also reduce the number of computations you can do and still, let's say, account for the mutations that we may have in a particular population. So this means that you may do your analysis maybe more accurately. By the way, you can ask your questions anytime. You don't have to wait for at any point, let's say. So basically, this is read mapping, where we've mainly covered basically how we use certain data structures and heuristics to figure out where a read belongs to. So then we use this information that we generate from read mapping for many reasons. You may either use it to construct the genome itself of an individual, or you may use it to identify the mutations in an individual that we call variant calling, or you may use it for metagenomics purposes, where you identify what sort of organisms that you have in an environment and what is their ratio compared to other organisms. So I'll start quickly with the assembly. I'll show you basically their high-level steps. So the question is, we've been talking about basically how we do the analysis given a reference genome, but then again, what if you don't have that reference genome, right? Or what if you don't know what sort of reference genome you need to use? So it can happen. And then for that, basically the computational step is to perform assembly, which essentially creates the individual genome from scratch without looking at the reference genome. And usually a common step to do it is once you sequence the individual genome and then construct the reads, these are essentially the fragments from the individual, what you'll do is that you'll try to figure out the overlapping regions between reads. So you figure out these overlapping regions, or also you look at the reverse complements, basically other strand information from the individual, and then you sort of construct, let's say, some overlapping information between reads. And then there are some other further steps where you, for example, remove some redundant information, you do some cleaning, and then you figure out the layout or the connection between the overlaps. And then once you figure out this connection, then you can perhaps take a consensus between these connections, and then you construct the assembly. So one way of doing this is actually, again, to use graphs. But again, this is not going to be as simple as this, but rather it usually looks a bit more complex. And then really, then you need to clean up this graph so that you can extract the useful information. Eventually, the assembly that you can construct can be erroneous. And if you want to correct the errors in it, you need to basically use additional tools. Here we describe a ML-based approach to correct errors that you may have in an assembly. We also basically describe how you can essentially accelerate the ML approach, again, to correct errors in the assemblies and also to do more things. Second, how do we find variations? Or what it means actually to find a variation in an individual. So once you map your reads to a reference genome, as I said, you're going to do the step that is called alignment that will figure out basically the exact differences between a read and the reference genome, right? So this could be perhaps maybe the substitutions, the small changes, or it could be the insertions or deletions that you may have in an individual. But they are not necessarily mean, or it doesn't necessarily mean that these differences are directly mutations that you may have in an individual. Rather, maybe you either map the read incorrectly, it's in the incorrect position, or maybe you have a sequencing error, right? So it's not necessarily a mutation of an individual. So then you really need to figure out what is a mutation or what is not a mutation, or maybe an analysis error, let's say. So to do this, we do variant calling, and in a high level, what a variant calling is, you align your reads, you map your reads to reference genome, and you sort of get a consensus of basically what each read is telling compared to what the reference genome is telling at a particular position. So this is basically the high level step that we're doing. And then in order to basically figure that out, you really need to basically do some statistical analysis, whether this is an error or whether basically this is even an artifact that you generated during the sample and library preparation step, where you basically, for example, amplify your read multiple times. You generate the copies of your reads multiple times, and maybe you're just mapping your read here maybe 100 times just because of that, the copies that you generated. So these are basically the high-level steps. So what sort of variants that we may have? One is something that we call SNP, or single nucleotide polymorphism. So these are like single changes that you may have, single mutations you may have in your genome. So we also have insertions or deletions. So you may have inserted characters, additional characters, or deleted DNA characters in your genome. Or you may also have structural variants. These are much larger variations that you have in your genome. And theoretically, they should be larger than 50 characters, if I remember correctly, to be considered as a structural variation. And also we have copy number variations, where you may have basically the multiple copies of the same DNA content many times in a repeated manner in the genome. So these are relatively simple problems to solve, and the structural variations are relatively harder problem to solve given the sequencing technology that we're using. People are also using ML approaches to identify these variations. Imagine you represent your alignment information as sets of images, and if your image is different than your, let's say, ground truth image, so how do you figure out whether this is a variation or an error or something? So this is basically what the deep variant tool is how it is encoding the problem, and then how it is trying to solve this structural variation, a variant calling problem. And so the last step is another downstream analysis step is metagenomics. So metagenomics, as I said, you have an environment of that includes multiple organisms, but essentially you don't really know what sort of organisms you have, right? So you have a bunch of DNA, and then your goal is basically to figure out what sort of organisms that you have. And then basically a common approach to do it is you again extract the reads, and then you extract the k-mers. And then what you essentially have is that you have a large database of, basically you have a large database that contains many organisms, right? So you may have a database of bacterial genomes, or maybe even all the genomes that we've figured out so far. So this is basically, as you can imagine, this is a huge information. And then what we can do is that then we can essentially query all these k-mers in that database to figure out whether we're basically matching a certain genome or not, and then estimate their abundance. So this is called relative abundance estimation, where you figure out, oh, okay, so in this sample you have maybe 55% of E. coli, a bacteria, maybe 20% of virus, and so on, right? And this is important to analyze because you really want to figure out whether, for example, there is a harmful pathogen that is in this environment. Maybe after understanding, you can take actions accordingly. So these are basically the downstream steps that we have in genome analysis. I'll basically move on to the steps to explain how we accelerate these steps in genome analysis. So we basically believe there are significant barriers to genome analysis. And basically, there are like multiple reasons for that. One is we essentially believe there's a performance gap between data generation and data processing. So essentially, there are multiple steps in genome analysis, as we covered previously. But essentially, the speed that we're generating data and also analyzing this data, it essentially is very different across steps, right? So then this means that it is very highly likely that you're going to be bottleneck in one of those steps, right? So then the challenge is, how do you figure out essentially what is the bottlenecking step, and then how do you basically unblock that bottleneck? And one big reason for that is, again, the ways that we analyze this data. So how do you essentially accelerate this particular step? And one big reason for this is the expensive data movements, right? So remember, I mentioned essentially there is this the data needs to move across multiple levels in the memory hierarchy. So this means that you're introducing lots of latency, let's say, in this process, even though even if you need to do very simple operations, right? Maybe you just need to do a simple addition. And even for that, you need to basically move this data from where it is generated across multiple levels so that this simple addition can be performed. So this is very costly, not just in terms of the performance, not just in terms of the latency that it causes, but also in terms of the energy. And especially if you're targeting, let's say, portable and in-field analysis, the energy is a big concern. So for that, you really need to reduce this data movement over here so that you can accelerate the analysis itself. So basically, the data is performed far away from the data, and then we need to basically close that gap eventually. So this means that we need to orchestrate the algorithms and architectures well to handle the data well. And then to this end, of course, we also release many materials on YouTube. So if you're interested in this direction, you can take a look at these materials. There are essentially many talks and lectures that we release in this direction. But I'll basically quickly cover some of the works that we do in this direction, and one of them is GenPip. So remember, again, I'll quickly show you the steps in the genome analysis, right? We start with the data generation, which is raw data, and then this raw data is stored. And then what you do is that you translate this raw data using a step that is called base calling. And then you essentially generate let's say the data in a form that is in human readable. And then you again store this data, and then you again do the computation. You do some computation, and then you move on. And then you again store, and then you do another computation to generate more insights and so on. So basically, what I'm trying to show here is that there's basically lots of steps here that performs computation and then stores the intermediate data and then does the computation based on that intermediate data and so on. So based on that, we essentially identify two problems. There are multiple steps in genome analysis. So this causes large data movement between multiple steps. And also there's lots of wasted computation that we do just because basically using a data that becomes useless eventually in the later steps of genome analysis. So how do you solve these problems, right? So again, I'll illustrate these problems again in a bit more lower level. So assume that you generate your raw signal data is basically relatively larger. You base call it, the data size reduces a bit. You do some analysis. Your data size reduces a bit further. You do again some more analysis, then your data size again reduces, right? So then what you're essentially doing is that between the analysis steps, you're essentially moving large volumes of data between the steps, right? So there's lots of data movement here. But also assume that you're starting with 100% of the data that you generated after base calling, right? After some step, some of them will be eliminated. You're not going to be using them in the later step. Again, after some computation, even a much larger portion of it will be eliminated again. They will be useless. So then this means that you're only basically using eventually a smaller portion of the data. So then this means that whatever becomes useless in the later steps, they are still computed in the previous steps, right? So this means that you're paying the cost for moving the data, but also doing the computation for something that becomes useless later on. So how do you solve this? You can, of course, accelerate certain steps, right? So that the computation is accelerated so that latency is reduced. And there are certain approaches to it. For example, you can apply a PIM approach, processing in memory approach, where you don't necessarily move the data to do the base calling. And also in the later steps, you can also do PIM again to reduce the data moment, right? So this reduces the data moment overhead, but again, it doesn't really solve basically the user's computation that we're doing in between these steps. So then we identify certain opportunities in this area. We do some motivation analysis to whether there's really an opportunity or not. And then to do this motivation analysis, what we first look at, essentially we assume there is a PIM accelerator to accelerate the base calling step and the read mapping step separately, right? And then we then try to basically figure out what sort of speed up that we can get compared to essentially to the analysis where you're not using these PIM accelerators. Then we also go one more step beyond, and then we assume that there is no data moment between the accelerators. So this is a bit, let's say, an opportunist, let's say, optimistic assumption, right? So it's not necessarily you're not going to have a data moment, but this is the assumption that we're making. And then based on that, we further identify that we can further accelerate basically this genome analysis step beyond these accelerators. And then we also assume that there is no data moment, but also there is no useless read. And by looking at that, we also identify we can even further accelerate this step compared to these two assumptions. So then what we really claim is that we essentially minimize the data moment between the steps, but we also minimize the useless computations that we're doing between these steps. And then to do these, we propose GenPip. So GenPip is really basically an accelerator that sort of combines the base calling, the step with the read mapping step, but also by also essentially figuring out what is whether computation will be useless or not later on, and it tries to figure that out as early as possible so that we don't do unnecessary computation in the later steps. So there are two steps in GenPip. One is something we call chunk-based pipeline, or CP. So what it does is essentially, I'll actually illustrate this instead of showing the text. So this would be your conventional pipeline in base calling. So what you would be doing is that you have this full raw signal, and then you would be base calling it chunk by chunk. And then once you base call the entire signal, then you start your analysis, which is maybe quality control and then the read mapping, right? Using the entire base call data. But then what you could essentially do is that after you perform your initial base calling, you don't have to wait for, let's say, the entire base calling process to finish. Rather, you can start your partial computation that you'll do here and here immediately as soon as you generate the initial information, right? And then by doing so, you can also figure out whether actually you need the further base calling steps or not, because the initial computation will also give you some insights, whether essentially the read mapping information that you're generating, whether it basically tells you that this read belongs to the reference genome you're mapping or not. Basically, if it doesn't belong to, you can even stop somewhere here so that you don't do the useless computation. And then this is basically what we call early rejection, by identifying these useless computation early on while doing the base calling. So this is basically the hardware design. I'm not going to go over it, but what it essentially does is that on the left, we have the base calling module, and on the right, we have the read mapping module. And in the middle, we're essentially controlling the data moment between them, right? So we're continuously base calling the data and then doing the read mapping as soon as a part of data is base called. And then based on that analysis, we're providing feedback again to the base caller accelerator back and forth. And then doing this, you sort of reduce the unnecessary computation and data moment. And then we essentially look at the results where we compare GenPip to CPU implementation of base calling and read mapping, also GPU implementation of them, and also an optimistic PIM where we assume there is no data moment, but there is still some useless computation. Doing so, we show that GenPip provides substantial speed up compared to the baselines, as well as significant, essentially, the energy reductions, because it doesn't only basically reduce the data moment, but also it reduces the user's computation. So this is all basically a processing in memory approach. If you are interested in learning more, you can take a look at this paper. We also focus on essentially how we can perform the base calling step using emerging technologies, for example, memory-centric devices, fully using analog computation rather than, let's say, using other approaches. And then these approaches are mainly based on the Kirchhoff's law and the Ohm's law, where you do the multiplication and addition using the analog computation. We also have other approaches where we use processing in memory to perform, for example, genomic profiling, where we identify whether there are harmful, let's say, pathogens in the food that you're analyzing. And also we develop or we publish other works where we use, again, processing in memory to perform the approximate matching, the alignment step. So I covered some ideas where we use processing in memory, but the next question is, can we also process the data closer to where it is stored? And for that, we've also published several works in this direction where we do the computation within the storage, within the SSD. And one good example of this direction is GenStore. So essentially what we're observing is that assume you have a storage system that perhaps includes a single SSD or multiple SSDs. And also in the hierarchy, you would also have a main memory, caches, and then your computation unit, right? Either CPU or an accelerator. And in the conventional, basically, system, what you would be doing is that you would be sending your data from the storage system to the computation unit so that it is processed there, right? And for example, if you're doing alignment is there, the alignment is costly. And there's also this data movement overhead where you need to move the data from the storage system to the computation unit. And of course, there are accelerators, right, or heuristics where we, for example, try to reduce the amount of data that we store in the main memory, or we design accelerators so that we accelerate the computational cost steps, right? So that, or we design filters so that we reduce the unnecessary computation, right? So then this means that you sort of mitigate the computation overhead in genome analysis. But as you do so, you reduce the computation overhead, but then actually data movement overhead becomes a bit more abundant. Assume previously you would be, let's say, consuming 30% of your entire time for moving the data, right, from the storage system to the computation unit. And then the rest of it would be done for the computation. But then you accelerate all these steps so much that now maybe that 70% becomes, let's say, 10%. Now, let's say you're actually, your major computational overhead or your major performance bottleneck is basically the data movement. So how do you basically minimize that? For that, then you need to not move the data from where it is stored, and rather do the computations near them. And you can actually do that. What you can, for example, do is you quickly figure out which reads that need to be sent to the computation units, and then which of them they shouldn't be sent because maybe they are going to be useless later on, or maybe you can even do very simple computations so that such that they are useful, but you've figured that out that they are useful doing very simple computations within the storage. For example, one example of such a simple computation is the exact matching of reads, right? So if you can quickly identify if the read is exactly matching to a reference genome, this means that it actually should map to that region in the reference genome, right? So you don't really have to basically do the costly alignment process and then move the data to basically the computations and so on. Rather, you can basically design basically a logic here that is extremely simple, that literally performs just exact matching, so that it figures out whether this read needs to be sent or not. And if there are basically non-matching reads, you can also do some filtering based on how many basically mismatching bases that this read has compared to the reference genome. And if basically the read is too dissimilar to the reference genome, you can also decide not sending the data to the computations. And this means that you're also not paying the cost of basically the data moment, which then reduces the latency. So basically, these are the two opportunities that we're leveraging in GenStore. And then doing so, you can essentially accelerate the entire genome analysis by co-designing, let's say, your storage system and the algorithms that you're using behind it. Are there questions? Okay. So basically, this is GenStore. I'll move on to the other basically work that we recently presented at ISCA. This is again a work from Nika, who was also first author of the GenStore paper. So Megas is mainly focusing on the metagenomics analysis. So remember the idea in metagenomics, where you're trying to identify the organisms in your sample. But essentially, again, the data that you're dealing with is huge, right? And as well as the database that you're using is huge. So basically, the idea is that can we somehow leverage the SSDs again so that we can do some of the computations that is needed for metagenomics analysis within the storage system, rather than again sending the data to the accelerators or to the CPU. So these are the steps. As you remember, we start with basically the k-mers from the read, and then you basically try to match these k-mers by querying a large database that contains the information on many species, and then you do some computations to identify whether this read belongs to any of these reference genome, and if so, what is the ratio of the organisms that you have in your sample, right? So basically the high-level operations that we do in this metagenomics analysis mainly k-mer matching, and then this is basically some sort of intersections, let's say, between the reference genome and the read, and also basically the query to the database, right? So then what you can do is that you can sort of co-design your storage system such that you sort of orchestrate, let's say, how you move the data from the host system, because it may so happen that some of the steps maybe just they are not so costly that you can maybe use CPUs and so on. But then how do you orchestrate the data between the host system and the SSD such that perhaps maybe you don't access the data randomly in the SSD? So then this means that you need to also prepare your database in a way that it doesn't require random accesses, right? So then the question is, how do you redesign that database also in a way that it will not require random accesses to SSD? And also, how do you some of the computations that is needed in the metagenomics? For example, the intersection step, right? So you could design maybe accelerators, right? And then it does the intersection, or maybe you could do basically the internal, the computational capabilities that the SSDs provide to do the intersection operations. So we analyze basically all of these opportunities, and then we look at basically how Megas compares to the baselines, the metagenomics baselines, where one baseline is performance optimized, meaning it performs the metagenomics analysis in a quick way, and also an accuracy optimized baseline where it performs the metagenomics analysis in a very accurate way. Looking at both, we identify that the Megas basically achieves significant speed up here, up to 15x compared to these baselines. We essentially also look at yes, I think this is what I just said in the previous slide. So we look at also different architecture baselines where you use also the PIM approach, also a performance optimized approach and accuracy optimized approach, and then also compared to PIM approach, we show that Megas reduces the energy reduction, reduces basically energy consumption compared to these baselines, essentially. So we also do some other analysis here where we also look at the area and the power usage and so on. So if you're interested, you can take a look at this paper. So this is Megas. We also have Genome. I'm not sure if I have much time left, but I'll quickly show you what we do with Genome. Genome is mainly basically an accelerator to perform computational closest step in genome analysis, which is the approximate sitting matching problem. So this is basically based on an algorithm that is called a BiTAP algorithm. So we sort of modify this algorithm such that it becomes a bit more hardware friendly, but also we sort of extend this algorithm such that it performs, it essentially provides you additional features that is needed for alignment. So this is basically how the Genome hardware design looks like. It has two parts. One part is distance calculation, or DC. The second part is the traceback, which is needed basically once you perform your approximate string matching. You basically then basically do the traceback to identify the mutations and the other edit operations. So maybe I'll just take 5 more minutes before we take a break. And then essentially the distance calculation part is nothing but basically simple logic design where it performs, where it identifies the mismatches and insertions and deletions given a vector of basically from the read and also in the reference genome. But what it also features is that there's a systolic array design, because what we observe is once you perform basically a simple step in approximate matching to identify whether there's a mismatch at a particular position, then you will move on to the next position to identify again whether there's a mismatch and so on. And then to do this, you don't really need to basically send your result back to the memory and then get it back again to do the next step of computation, right? So you can actually flow this, you can essentially design a systolic array where you flow this information across multiple processing elements so that you sort of eliminate the data moment between them. We also do traceback. So traceback is not originally included in BiTAP. This is one of the contributions in Genome as well. So we do the traceback so that we can identify, we look at basically what we generated previously in the distance calculation step, and then identify the actual operations, the insertions, deletions, and so on, so that you can identify the mutations eventually. So if you're interested in learning more about Genome, you can take a look at these. Scrooge is basically an addition to Genome where we improve the capabilities of Genome, and also we provide implementation for the CPUs, FPGAs, and the other architectures. Segram is an extension to Genome as well, but it essentially looks at the problem in the graph context. We sort of encode the problem in a graph data structure and then extend the Genome such that it can perform the mapping given a graph structure. So you can take a look at this as well if you're interested in. We also work on basically designing algorithms, not just architectures, for genome analysis. For example, one question is, what if basically the reference genome gets updated, right? Do you really need to do the computation from scratch? Or can you somehow leverage the information that you generated previously by looking at the previous reference genome, right? So the question is yes. And we leverage this information so that we reduce basically the amount of computation that you need to do whenever there's a new reference genome. We also look at newer sequencing machines and then try to identify what sort of opportunities we have there. So here, this work essentially asks the question, do we really basically need to translate the raw sequencing data, for example, we generate from nanopore reads, or can we directly basically use these electrical signals to do the genome analysis? This means that you essentially eliminate the translation step, which is a computational cost step, and then you can do your analysis much more efficiently, maybe even a more accurate way. So these are the steps basically in the raw hash work to do this analysis. So I'm going to skip these relatively quickly, but the slides are available. You can take a look at these if you're interested. Of course, we continue work on this direction. We publish more works to make this type of analysis more accurate, but also to enable more applications. For example, here, we look at the problem in a way that actually can we identify the mutations within the electrical signals so that maybe you can even do your variant calling, let's say, even without performing base calling, right? And in order to do it, you need to do alignment. And in this work, we do the alignment and then show that we can actually identify perhaps mutations for the future work. And even so, we try to perform assembly. We try to construct basically the genome from scratch without looking at the reference genome and without even translating the signals to bases. And doing so, and how do we essentially do it? We essentially try to find similarities between electrical signals so that these signals then so that they can be converted to basically the genomes, the individual's genomes somehow by looking at the overlaps between them. So we believe that there are many new applications, there are many new use cases based on these new technologies and new architectures and so on. And of course, the industry is catching up. They are also looking at these architectures. For example, this is the Dragon FPGA that is now essentially integrated in the Illumina sequencers. So when you, for example, buy the sequencer, it will contain an FPGA that accelerate some of the steps that we do in the genome analysis. So essentially, the biotech companies are already looking at these solutions, but also it's not only biotech companies. Also, the industry, the tech companies are also trying to provide solutions for important workloads, right? For example, if you have so many DP operations in your analysis, as we saw happen essentially in genome analysis, can you basically provide a support for that such that your DP operations are going to be accelerated, for example, right? And in this H100 architecture, there are these basically specialized architectures so that your dynamic programming computation will be faster. So that whatever the workload that is based on these dynamic programming operations will then eventually be accelerated. Biotech companies and also tech companies are collaborating to accelerate their steps. But also there are like other steps in genome analysis, right? For example, some steps relying on AI ML operations a lot. And essentially we have these basically, for example, wafer scale engines, right, that accelerates the AI ML operations a lot. So this is the Cerebras chip from 2024. Basically contains a large amount of cores so that you can accelerate the AI operations compared to the other architectures, for example, the state-of-the-art GPUs. And also you can use processing in memory to accelerate your applications, as I covered previously. And we are also part of this effort in a project that is called BioPIM, where we have many partners from the industry and also from the academia, where we are basically trying to co-design algorithm and architecture such that so that we can apply the PIM architectures to accelerate the genome analysis. So if you're interested in all of these efforts that we're putting in our group to accelerate the genome analysis steps, you can also take a look at these materials and learn more about it. We also organize workshops joined together with the top conferences in the genome analysis field. And also we have courses that we're offering where we teach basically the fundamental steps in genome analysis. And also we essentially propose projects and then mentor students if they are taking these courses so that they do some hands-on projects for genomics and to accelerate them. So to sum up, I think this is an exciting time to look at another important problem, right? It's not basically AI, ML, but another exciting approach, another exciting direction is genomics or health. And I think this is the right time to look at this. Essentially, how can we build algorithms and architectures, and how can we co-design them together so that we're not only basically accelerating those, but we're essentially enabling new applications that were not possible before. So if you're interested in these directions, feel free to contact us. With that, I guess I'll stop so that you can take a break, but maybe before that, if there are any questions, I can try to take them. I guess this was a very dense, almost 1 hour lecture. But I guess you can also contact us, I guess, later on if you have questions. Okay, I guess then we can take a break for maybe 7 minutes. Is it fine?
Maybe until 5:20.
5:20, okay, good. Good, then let's come back in 5:20. Thanks, everyone.
Is there a reason why people like that side of the room more? We have an load imbalance problem here. I'm not sure if it's a problem, but it's definitely load imbalance. Is the Wi-Fi better over there? No. It's almost 22, so we'll get started soon. Everything's okay online? Okay. Okay, let's get started. John gave a very fast overview of genome analysis. Some of the things that he was discussing at the end were actually quite exciting things like raw signal analysis. How can we rethink the genome analysis pipeline without doing translation from these raw signals that come from devices to text, but directly operating on raw signals? There are a lot of interesting algorithmic issues over there, as well as issues to accelerate those new algorithms. So if you're interested in any of those, feel free to contact us. It's an exciting area. We're doing a lot of work on raw signal analysis these days. And going forward, we're even though his talk was focused on genomics, there are a lot of issues in general in omic data, like proteomics, protein analysis, and other types of biological sequence analysis. And this data is growing and growing. It's not going to reduce anytime soon. It's going to grow faster and faster as technologies become better and better to produce this data. So there's an increasing need for like both better algorithms as well as better architectures at the same time for this sort of thing. But I'm not going to talk about that. I'm going to switch gears right now to give you a perspective on a little bit different area, which I think also is quite exciting. And we're doing work in this area also. We're not going to be able to cover all of these slides, and I'm not going to rush. So you can study the slides on your own. But feel free to ask questions also. Rahul can actually cover some of these in a later lecture, maybe, if that exists. But we're going to start presentations next week, so there are no more introductory lectures. So I'm looking forward to you guys presenting as opposed to us presenting. And I think we're going to have fun covering a lot of interesting papers this semester. Are you guys ready? Excited? Who's presenting next week? Okay, only one. There should be two, but maybe the other person is not here. Okay, which paper are you presenting? RowHash. Okay, yeah. John didn't give you a lot of clues today. He just went over the paper, but hopefully you're finding it exciting. Yeah, I think it's really like we'll talk more about genomics in next week then. I think there's a lot of exciting things going on right now in genomics. Okay, so let's switch gears. I'm going to talk about data-driven architectures. Another way of thinking about it is ML-driven, since everybody likes ML word. I'll use ML over here. But it's really data-driven architectures that can basically become better over time. And specifically, we're going to focus on ML-assisted memory and storage management. And we call this this is one of the major directions that we've been pursuing. Essentially, we also call it self-optimizing architectures, as you will see. But basically, the problem is if you design systems and architecture, especially hardware today, a lot of the decisions you make are human-driven. Essentially, humans, some architect or some designer, designs the policies, and they basically essentially dictate how how one particular component should operate, what kind of decisions it should make under what conditions, essentially how to do things, right? But this may not be easy, and this is in general not so easy, as we will see in some examples, and because problems may be very complicated. For example, when you're designing a memory controller, you need to make many decisions. Even if you need to make one decision, which is scheduling which request to select to schedule next. There are a lot of considerations that you may need to make because you need to take into account the state of the system, state of the DRAM system, as well as state of the controller, as well as state of the cores. So it's very difficult to design policies that are really optimized, that work well in many conditions, many different workloads, and workloads may change over time also. So as a result, what we have in hardware usually is many simple, usually too simple, short-sighted policies. These policies may not coordinate with each other also across different components. And essentially, there's no automatic learning that's going on. So if the memory controller is designed 5 years ago, that memory controller still takes the same decision under the same conditions. It hasn't learned anything, basically. It's a very dumb system if you think about it, right? And these memory controllers were designed probably 8 years ago or so, let's say, and they're still doing the same thing, even though they've seen a lot, probably. It's very different from how humans operate, hopefully, at least in the ideal world, right? Humans should become better based on experience. It's not clear if that always happens, but that's the goal. And at least we have the capability to do that if we actually nurture ourselves in the right way. And essentially, there's almost no learning. You cannot take lessons from past actions. So basically, the question we ask is, can we design fundamentally intelligent architectures? I don't know what fundamentally intelligent exactly means, but more intelligent architectures, at least, that can learn from their actions. So this is the vision, basically. We want data-driven policies, meaning machine learns the best policies over time, how to do things. It can change its mind, and it can become better over time, hopefully. So you buy this computer, and over time, it can figure out how to do things better in all of its components. It can adapt to the workloads that you're executing, for example, and maybe it gets rid of the inefficiencies in its policies. So as a result, hopefully we'll have sophisticated, workload-driven, changing, far-sighted policies, or policies that are not myopic, meaning if you look at a memory controller, again, I'll pick, one decision you make is to decide which particular request to schedule. Usually, you don't consider the long-term impact of that because a human designed the policy, and there's not an easy way for a human to decide the long-term impact of the scheduling decision. But if you actually design a learning-based policy, you can potentially steer it toward making long-term decisions, as we will see briefly with some of these. So basically, we want automatic learning, and hopefully all controllers will over time become intelligent data-driven agents that can coordinate with each other. So this, of course, requires rethinking the design of all controllers in this, with this mindset. So I'm going to show you multiple results that we have. We have looked at this perspective a long time ago. This is a paper that we published in June 2008 at ISCA. I'm going to talk about that. But then if time permits, we're going to talk about multiple other works. And Rahul did some of those works, and Rakesh was part of some of those works, so they may actually chime in. This room is really terrible. I have to look at the sun. You guys will experience this also when you present. Okay, maybe I'm not going to look at that thing. Okay, this is Rahul's work on memory. Basically, we first applied some of these principles to memory controllers, and more recently to prefetching. Another form of prefetching, accelerating long latency load requests by predicting which ones will miss in the caches, and also storage systems. So this can be done purely in software, actually, this particular mechanism, because the decision that you make in the storage system to place data is not really something that's extremely latency critical. So you can tolerate spending some more time to make that decision better. Whereas some of these other decisions that we're going to see for like predicting whether a request will miss in the caches, or what to prefetch, or what to schedule next, those are actually quite latency critical. So we're going to embed machine learning inside the hardware so that we can make those decisions. Does that sound good? Okay. Okay, so the broader vision is actually in a paper where we argue fundamentally better architecture should be data-centric, which we kind of covered earlier, data-driven and data-aware, which I'm not going to cover right now, but there might be some papers. Okay, let's jump in. So we were working a lot on DRAM controllers, and I like DRAM controllers, so that's actually a fascinating area. It's one of the toughest parts to design in a system. And this is not an easy part of the system. It's actually one of the hardest parts to design. How many people know the name Chuck Thacker? Nobody? How many people know about the Alto system? These are early personal computers. Okay. How many people know about Western Research Lab, for example? These are places where early computing systems were developed in the 1950s, 60s. And Chuck Thacker was one of the early people who worked on some of these early personal computers. And he won the Turing Award sometime, I don't remember when, 2009, I think. And I worked with him at Microsoft. Well, not exactly with him, but we talked a lot when I was at Microsoft, because he was at Microsoft at the time also. But he was actually into FPGAs at that time. And he was actually interested in building like what's called an emulation engine to essentially to accelerate system level simulation using FPGAs by offloading some of the things. And he had to design a DRAM controller to be able to do that. So he was designing a DDR3 memory controller. And we were doing a lot of research on memory controllers. And he was saying that's the worst thing or hardest thing he has designed in his life. And this is a person who started doing some of the earliest personal computers, which are clearly sophisticated. But DRAM controllers are much worse, basically, to him than those PCs. Okay, well, why is it difficult? Essentially, you have a lot of things to keep in mind. It's a very multidimensional problem. You need to ensure correct operation. That by itself is not easy because there's so many timing constraints, as we will see. We're not going to see all of them, but you need to do refresh at the appropriate time. You need to obey the timing constraints. On top of this, while obeying all of those, you need to service DRAM requests. And there are a lot of constraints over there. You need to keep track of all of these resources like banks, buses, channels, and different types of timing delays. And at the same time, translate requests to DRAM command sequences. We're not even talking about performance. To be able to do things correctly, you need to do this. To get performance, you need to buffer and schedule requests. You may need to do reordering, for example. You need to keep track of the row buffer. That's part of DRAM bank. Because if you actually have data that's in the row buffer that you can access, that's much faster. And then you need to basically manage all of these resources. And on top of this, you may need to provide quality of service, predictability, which is actually much more true today than in the past. And on top of this, you may need to manage power consumption and thermals in DRAM to make sure you're not really running out of your power budget. So there's a lot. We're going to focus on performance first, not quality of service, but later works cover performance. So one of the reasons why these are difficult to design is there are many DRAM timing constraints. So DRAM is controlled quite tightly by the memory controller. Memory controller dictates everything the DRAM chip does. As a result, specifications specify every single timing constraint that happens between different commands that you can send to DRAM. You can argue this may not be the best way of designing things, and I agree, but that's the way that has been happening for 20 years plus with synchronous DRAM. And there are many timing constraints. 50 doesn't do justice to it. It's really more than 100 in many DRAMs today. So just to give you an idea, there's a timing constraint between that you need to obey. That's the minimum number of cycles you need to wait in the memory controller before issuing a read command after a write command is issued. So you can do write to DRAM, and then you need to wait for some time to issue a read. This is because the bus is kind of unidirectional. You need to turn around the bus, at least at a high level. That's a good explanation. And then there's another timing constraint, T, row cycling time, which is the minimum number of cycles between the issuing of 2 consecutive activate commands to the same bank. If you activated one row, and if you want to activate another row, you need to wait for some time. That's the row cycling time. And that's usually pretty long. And then there are many others, of course. On top of this, you need to keep track of many resources to prevent conflicts, to keep track of timing constraints. There are many different timing constraints there. As you can see there, so write to read delays are, for example, a rank level timing constraint, meaning it affects all of the chips on the rank, whereas TRC is a bank level timing constraint. So you need to actually keep track of all of these different resources and what's happening in all of those resources. You need to handle DRAM refresh to retain data, manage power consumption, we've already discussed this, and optimize power, performance, and quality of service on top of all of this. And reordering is not simple. And if you want to provide fairness and quality of service to different applications, that complicates the scheduling problem. Here are some examples from one of our early works. It actually has a nice idea over here. The idea in this work is actually whenever you're doing evicting something from your last level cache, consider what's going on the DRAM side. If there's a row buffer that's open, evict some things proactively from the cache so that you can actually write them quickly to the DRAM, as opposed to evicting some things randomly, which causes row buffer conflicts. So that's an example of last level cache and memory controller co-design. And I think it's a kind of nice idea. But for this, you need to also consider some of the timing constraints, as you can see over here, but I'm not going to talk about that. If you want to learn more about timing constraints, these 2 papers actually do a pretty good job, in my opinion. I'm not saying that because I'm a co-author of them, but one way of learning about timing constraints is to go and read data sheets by IEDM manufacturers. That gives you something, but it doesn't give you any insight because they don't tell you a whole lot as to what is what. And in some cases, they're very cryptic. These papers actually provide insight into why some of those timing constraints exist and what is their scope, etc. For example, act pre, etc. But you can read the paper for more detail. Okay, so that's it. That's some of the complexity. So people have designed a lot of scheduling policies. Clearly, you need to decide what you have a buffer in the memory controller, and you get requests from one or more processors. Sometimes even the DMA engine, storage engine, for example. GPUs, different kind of accelerators. And how do you decide which request to schedule first? One policy that's the simplest is FCFS, first come, first serve. There's no scheduling over here. You just schedule the oldest request that has arrived, right? A bit more sophisticated is what's employed in many memory controllers, a form of it, a more sophisticated form than what's described over here, which is FRFCFS, first ready, first come, first serve, where you prioritize a request that goes to a row that's already in the row buffer. So you need to check the row buffer for this, and otherwise you prioritize the oldest request among all of the equal ones. The goal is to maximize the row buffer hit rate, to maximize DRAM throughput. So on top of this, of course, you need to think about existing memory controllers need to think about write, read, right? Because you cannot do reads concurrently with writes because of this write to read and read to write delays and the fact that the bus operates only in one way. As a result, there's also another policy over here to decide when to schedule writes and when to schedule reads. So think about that in the back of your mind. So things get complicated. But then you can do more sophisticated policies. You can prioritize based on request age, row buffer mid status, request type, read writes I mentioned, but there's also prefetches. Requester type, whether the request was generated by a load or a store. This may be important because if it's generated by a load, it may be blocking at some point the core because it's a load, it's loading some data that's going to be needed by some other instructions. Whereas if you're doing a store, you may not need that data immediately, right? So you can actually buffer the stores and it may not have dependence close by in the instruction stream, right? And then there could be policies based on criticality, but for some measure of criticality. For example, is this request belonging to the oldest miss in the core? Is it going to is it at the top of the reorder buffer, meaning it's going to block the instruction window, or it's already blocking the instruction window? How many instructions in the core are dependent on it, etc. So you can imagine all of these things, right? And then you can also take into account other metrics like what kind of interference is this request causing to some other cores? Because if you're running together with multiple cores, then this request may be causing interference. So you can imagine a lot of policies, as we have kind of done in our research. I like this area, and I'm not going to talk about any of these, but these are all heuristic-based policies from our perspective, at least. This is one of the earliest works that we've done, where we've shown that if you actually don't do careful scheduling of requests, one application may deny service to another application. And then we try to solve problems like that. It's not just a denial of service problem, of course, it's also a performance problem and quality of service problem. These are coupled with each other. So we've written a lot of papers. This is one paper that's interesting. It's implemented in some of the SOCs even today. And there are other papers that we have written on the topic that I'm not going to go over. If you're interested, you can take the computer architecture course and you may cover some of these. And then there are more difficult problems when you actually combine CPUs and GPUs together. Then actually you have many different cores essentially competing for service from the memory system, and you need to take into account the quality of service requirements of those cores, because GPUs may have some frame rate requirements, for example, and if you don't satisfy that frame rate requirements, you actually make the user very uncomfortable. CPU applications may or may not have different quality of service requirements. So actually designing a policy taking these into account is not easy. And this is one of our latest papers that looks at CPUs, GPUs, and other hardware accelerators like audio and video accelerators. That is interesting. So if you're interested in this area, there are a lot of heuristic-based policies, and this is one of the most sophisticated heuristic-based policies. And actually, people employ this sort of thing in real SOCs today. Real SOC memory controllers need to take into account all of these things. And then there's also predictable performance. If you really want to predict the performance of an application, we've developed methods for estimating slowdowns, for example, in these works. So my takeaway is, I think memory controllers are really critical to research, and they will actually become important, more important. The question is, are we really designing them in the best way? So this is one picture to show you that how things are becoming more complex. So you have general purpose cores, accelerators, and GPUs, and they're essentially all accessing main memory together. Today, things may be separate, but really the fact that they're separate is a bad thing. If you have CPU and GPU memory completely separate from each other, you have to move data between the CPU and GPU, for example, and that's not necessarily a good thing. But this is happening or going to happen, or in some cases already happening, in even worse than what I depict in this picture. So you have heterogeneous agents accessing memory, and there's main memory interference, and there are many goals, many constraints, and many metrics. I've kind of talked about some of them. Okay, so we're not going to tackle that general problem, but this is just to motivate that there's, so if you actually look at this, and if you want to design an optimal policy, let's assume that we can define optimal, which is not easy to begin with, then as a human, I feel lost. Even though I worked a lot on memory controllers, I don't know how to do that, basically. It'd be nicer if we can actually have a domain expert who knows a lot about memory controllers to figure out and somehow embeds a policy that's actually much more rigorous. And that's why we actually turn to machine learning. Basically, the reality is that it's very difficult to design a policy that maximizes even one metric. Forget about 2 or 3 or 4. We're going to focus on one metric, which is performance. There are too many things to think about. Thanks, Rahul. This is Rahul's picture, not mine. So on top of this, there's continuously changing workload and system behavior. Let's assume magically you found a policy that's optimal with one workload. You could potentially do that with one workload, even though a workload can be actually very heterogeneous in itself. But now if you go to another workload, that policy may not be good enough. And then if you go to a slightly different system, meaning if you allocate the resources and the caches slightly differently, maybe that workload also will change in terms of the pressure it inflicts on the memory controller. So basically, there's just too much to think about and too many dynamic variables that change. So our dream was to ask the question, wouldn't it be nice to have a DRAM controller that automatically found a good scheduling policy on its own, as opposed to the designer figuring out what that policy is and dictating it? And clearly this has advantages because hopefully that policy will become better as well and dynamically adapt to what's happening. So that's the problem, basically. Yeah, I think I've already said this. It's difficult for human designers to find that policy and design that policy. And the idea is to have a memory controller that adapts its scheduling policy to workload behavior and system conditions using machine learning. And in this case, we found out that reinforcement learning maps nicely to memory control. It's not a neural network, it's really reinforcement learning. Although you could use neural networks in combination with reinforcement learning, like deep reinforcement learning today. Essentially, the idea is to have a reinforcement learning agent inside the memory controller to do scheduling. Basically, by doing so, you can dynamically and continuously learn and employ the best scheduling policy, and you can hopefully maximize the long-term performance. I'll briefly talk about that. So what is reinforcement learning? Essentially, in its general form, a reinforcement learning agent interacts with its environment by taking actions. And after taking actions, it observes a reward based on that action. And that reward function needs to be designed. How many people know about Pavlov's dogs? Okay, some people. Yeah, this is basics of really reinforcement learning, psychology, right? Ivan Pavlov figured out that you could train dogs to react to bells by every time you ring a bell, you put food also. And then next time you ring the bell, the dog drools, even though the food is not there anymore. So they associate basically the bell with the appearance of food, and then they take an action, which is drooling in this case, or maybe appearing when they were not there. So that's a reinforcement learning, in its purest form, right? Essentially, the agent observes the state and takes an action, and hoping that based on that action, they will get a reward, right? This way, you can actually train the dogs to do many things. And Skinner trained pigeons, for example, to deliver messages in World War II with this approach. So you can actually train a lot of animals this way. Humans are also like that. Basically, we are reinforcement learning agents, at least partially. I don't know about the fully, but partially we're reinforcement learning agents, right? It's at least the basic things we learn relatively quickly with reinforcement learning. If you touch a stove in a given state, you learn not to touch a stove quickly because you get a negative reward. Or you don't get a reward, maybe, I don't know. I don't know what kind of reward you expect when you touch a stove, but that's beside the point. Okay, so if you apply this concept to a memory scheduler, the memory scheduler observes the state of the system. It could be the memory controller, it could be the cores, it could be anything in the system, actually. And then, based on that state, it takes an action, and based on the past experiences. And it observes a reward. In this particular case, we specify the reward as data bus utilization, but it doesn't have to be. It could be some other metric of performance because we're trying to optimize performance. And over time, it figures out what action to take in a given state to maximize long-term data bus utilization. That's the idea. That's the goal over here. Now, clearly, you can change. So what is in control of the designer right now? The designer decides what actions are available. I mean, the designer actually doesn't have a whole lot of choice over here. They could add or remove a little bit, maybe, if they're not like critical, but these are more or less given the DRAM interface, right? We know what actions are possible. You refresh, you can pre-charge, you can activate, you can do read, write, etc. So it's a limited set of actions, clearly. So nothing changes over here. State is important. Basically, what kind of state attributes do you examine? What is the input to this mechanism? And reward is, of course, defined by the designer also. In this case, as designers, we chose data bus utilization because we wanted to maximize that, because we were actually looking at parallel workloads. And data bus utilization is not a terrible correlator to performance. If you maximize the data bus utilization, you're improving the throughput on the DRAM bus. And if your workload is actually memory bound, improving throughput directly translates to performance. Ignoring some second order effects in these workloads like interference, etc. So we didn't want to deal with that, but later work dealt with that a little bit. So you can also choose to maximize a long-term reward. So you don't look at just the immediate data bus utilization, but you look at the reward that you get over time. So what was the data bus utilization in the prior cycle? Or prior time I made a decision, prior time I did a decision, prior time, and you weight them in a different way, so that what you try to maximize, the reward that you try to maximize, is a function of many decisions that are made in the past. Does that make sense? So it's really not the immediate reward that you're getting, but whatever you're getting over time. Okay, so how do you do that? Basically, we're going to use very basic reinforcement learning principles. And if you want to actually learn about reinforcement learning, there's a beautiful book out there. It's completely online. You can get the PDF of it. It's from Sutton and Bartow, Richard Sutton and Andrew Bartow, who are experts in this field. Richard Sutton actually is one of the early people who worked a lot on reinforcement learning. He has a lot of opinions on reinforcement learning also. But basically, that's a beautiful book. And we're going to employ a particular type of reinforcement learning over here called Q-learning. Essentially, what reinforcement learning does is it associates system states and actions, in this case, commands or actions, with long-term reward values. Basically, you take an action at a given state, and you learn the reward. You essentially learn associating state action pairs with a reward, right? And you keep this in a table. And over time, you update the rewards associated with given state action pairs, okay? And whenever you're doing a scheduling decision, you have a list of available actions, right? Not all actions are clearly available, but you know the state based on the state attributes. You hash the state to some, let's say, value. And you know the action that you may want to take. And you query this table using that state action pair. And for each state action pair, you get an estimated reward value based on what you've seen in the past. And you pick the action that gives you the highest reward value. That's the idea. And over time, if you made the wrong decision, you will not get reward, again, in that state of giving that action. So you will get negatively reinforced, meaning your reward values will reduce over time. So you can actually learn doing something else over time. assuming that action didn't lead to data bus utilization, for example, either, well, in the long term, based on that function. Okay, hopefully that's clear, right? And exactly how you do this is in the paper, etc. There are update functions, etc., that are in the paper. And that's the paper. Basically, if you look at the system scheduler observes a state, takes an action, and gets a reward. So we wanted to make it simple. So of course, having more sophisticated reward functions is also interesting. So what did we do? Essentially, this is the reward function. As I said, the goal is to maximize long-term data bus utilization. You get +1 for scheduling read and write commands. And then the update function takes that information as well as previous rewards into account. These are the state attributes. So how do you decide which state attributes to input so that you hopefully get something reasonable? This is one of the harder parts. This could be automated actually, but essentially we came up with hundreds of different state attributes, maybe more than 200, that we imagined could potentially be important for the memory scheduler to look at to make a good decision. For example, one of the attributes that eventually we selected is number of writes in the transaction queue, in the memory request buffer, basically, the number of writes, reads, and load misses. These turned out to be good. Good in the sense that we did feature selection, meaning you come up with 200 attributes and then you try to reduce the number of features or state attributes you want to use over a lot of simulations, essentially. That's how you figure out which state attributes are good. Potentially, you could do this dynamically, but it's a lot of work to do it dynamically and could be automated. Some of the state attributes that are interesting are, for example, number of pending writes for the reference row. If you're trying to access one row, for example, how many writes there are. This actually helps, in our opinion. Well, this was selected after feature selection because probably it helps with the read to write scheduling, for example. And also the reorder buffer heads waiting for the reference row. This essentially what's a reorder buffer head? Basically an instruction is waiting for the reference row and it's at the head of the reorder buffer. It's the oldest instruction in its core. And if that number is more than 0, that means something. If that number is more than 1, that means something else maybe, right? So more cores are waiting. And also, as you can see, request relative order in the reorder buffer. Like where is it in the instruction stream? This happened to come to the top, well, top 8 at least, state attributes according to our feature selection. Does that make sense? So there could be better ways of doing feature selection, but this is what we did. And then the actions are relatively easy, as you can see over here. We handled refresh of the with a separate mechanism. So refresh is not dealt with because it's a correctness thing. You don't want to mess with the refresh. Whenever a refresh comes, prioritize the refresh, basically. That comes in. But you can see that no op is action also. You can actually take no action. Sometimes taking no action may be better because you may want to keep the row open for some time. And the only action you can take is closing the row. And if your policy somehow learned that keeping the row open because something else may come, then you may take a no-op action. This is something that's not normally considered when you design a memory controller. As a human, at least the memory controllers I've seen and memory controllers I've designed, you always take an action. If there's a valid legal action to take, you always take it. You don't consider, you don't look too much into the future. But you could potentially do that also, right? Okay, but then it becomes even more complicated to design things as a human. Okay, I'm not going to go through the details of it. The paper actually has an implementation of it because implementation is important. How do you implement this in hardware? And we have a pipelined implementation, and there's a table that you need to store. The table is relatively large, it's 32 kilobytes, if I remember correctly. And there's also issues like how do you index into the table? How do you hash into the table? How do you make sure you can generalize? For example, the state attributes should not be taken very fine-grained. If you're very fine-grained, for example, number of reads, do you distinguish between number of reads 1, 2, 3, 4, 5, 6, 7, 8, 9, 10? Distinguishing between all of those leads to actually a very big table, potentially, or a small table or fixed size table that actually has a lot of redundant information. So one of the issues that you run into in general in machine learning is generalization or reinforcement learning. How do you generalize? So those issues are handled all in the paper by the design, and usually hashing and quantization helps these things. Okay, in the end, these are the results that we showed. Basically, on these parallel applications, we see significant improvements over FRF-CFS, which is the best baseline. So you see that there's 19%, and the headroom is really up to 70%. And we're not really using the most intensive applications. So the headroom actually goes larger. Headroom means this is an optimistic policy. If you eliminate all of the constraints except the data bus conflict, meaning you actually need to transfer the data, but all of the other timing constraints are eliminated. So we get pretty good, I think. And the performance improvements increase a little bit when you actually increase the number of channels, etc. Yes?
So I have a practical question.
Sure.
How programmable are these controllers? Because
Mm-hmm.
this would first allow reusing old systems, and it's also necessary to swap out policies.
Yeah. I mean, you mean the controllers we designed, how programmable are they? Or the controllers that are out there today?
Well, I imagine you would need in the future programmable controllers, but the current state is what?
Not programmable at all. Yeah, today's controllers, I mean, they don't even do this yet, but they're not programmable, basically. They're very fixed policies. So, for example, FRF-CFS that I mentioned, it's a very fixed policy. There could be something that you could slightly program, but at the BIOS level. You can basically cap the number of reorderings, for example, that you could do. You can specify some timeout, like how long you keep a row open, but that's the level of programmability. You cannot program the scheduler, but that's a good idea. Programming a scheduling policy is also interesting. Like you can, for example, it'd be nice to execute a program that does the scheduling for you in the memory controller, of course, in an optimized way, right? And people have proposed that idea also. But if you have this sort of policy, you could actually program the essentially the Q values, the reward values in that table, and that could actually help also. Okay. So, okay, I won't go into a whole lot more detail. There's a lot more in the paper, and we don't have any time, so I'm not going to go into the whole stuff also. But let me just give you the upsides and downsides, and then you can read the other papers on your own. I don't know if we're going to cover any of those papers. But essentially, the biggest benefit of this sort of policy is continuous learning, I think, one of the biggest benefits, in the presence of changing environments. Because now, hopefully, the policy that you design takes care of figuring out what's the best thing to do. In the paper, we discuss how we try to converge. Of course, we cannot prove anything because the system is very complex, right? If you look at the reinforcement learning theory, based on some assumptions, you can prove things. But we cannot prove this because this is very like dynamic system. But it seems to work, at least based on the results. And you can the other benefit I see is you reduce the designer burden in finding a good scheduling policy. Now the designer doesn't need to find a good scheduling policy. They just need to find what are the good system variables that could be useful for the hardware to find its own scheduling policy. And they also need to find the reward function, which is what target to optimize, but not how to optimize it. So it kind of takes, I think this is kind of the role of the machine learning. The designer still needs to do something important based on their domain expertise, but the really difficult part, hopefully, will be done by the machine learning, right? So it doesn't take away the designer's job. If you're concerned about that, I don't think that's ever going to happen anytime soon. Yeah, at least not with this sort of machine learning. But of course, there are difficulties also, right? How do you specify different objectives? So we just looked at performance. If you look at fairness, quality of service, energy, this becomes more and more complicated. But this is a good direction to think about also. Hardware complexity is always a concern, but maybe the benefits justify the complexity. Complexity is always a double-edged sword, right? You may, without cost, you cannot get to a better way of doing things. But it's still good to keep in mind. Maybe one of the most difficult parts is really the mindset and flow, because people don't think this way, right? People normally design a policy, they test it. So testing and verification flow is very much based on the policy, right? You know the policy and you know how it will react, and you write your test cases to figure out how it will react, and you test. So testing needs to change a little bit over here, because you cannot do that level of testing, right? At a high level, at least. You cannot say, okay, under these conditions, this is what I expect. Well, it's going to be a bit difficult given this. So you'll need to have a different approach to testing. You can test individual components, of course, but high-level testing is a bit more difficult. So this is the paper. There's a 2-page retrospective after 15 years, so you can take a look at that too. And there's later work that's improved on it to make it more quality of service oriented. And I will finish with this one. Any questions? No? Okay, then we'll start next week with your presentations. Looking forward to it. See you next week.